Constituent factors observed in viral diseases. Part 1 of 5. Pause text and illustrations for detailed study. Viruses are submicroscopic infectious agents that do not possess an independent metabolism. Viruses are acellular entities having no cellular structure. A virus can only reproduce entering a living host cell, using the host's genes to replicate itself. Viruses cannot generate energy. Since viruses cannot independently reproduce and are not comprised of cells, they are not classified as living cellular organisms. They are inert outside the host cell. In the absence of a host cell to infect, a virus is incapable of proliferation and will eventually cease to exist. Human genome to genes illustration. Viruses are microscopic units comprised of protein and the two types of nucleic acids, deoxyribonucleic acid DNA and ribonucleic acid RNA. All forms of life can be infected by specific viruses. The term genetic code refers to the specific sequential arrangement of amino acids from which proteins are synthesized. Viruses possess very small genomes. A chromosome refers to the thread-like nucleic acid and protein structure in the nucleus, carrying genetic information in the genes. Viral genomes do not encode for DNA replication mechanisms, metabolic pathways, or signaling molecules. They encode instructions for the translation of a small number of essential functional proteins. The nucleic acid genomes of viruses are based on the same genetic code as the cells of all living creatures. This accounts for the ability of viruses to reprogram and use the biochemical machinery of the host cell to support their proliferation and release to infect other host cells. This is a parasitical process. The incubation period extends from the time of exposure to the viral particle and its implantation, dissemination, and viral replication in the target cell, causing infection and subsequent viral shedding to other hosts. No signs or symptoms of disease are evident during the dissemination stage of viral infection. Basic scheme of virus illustration. Three types of subviral infectious particles consisting of genetic material or protein exist as smaller and simpler elements having some transmissible viral properties within the virus genome. 1. The human prion protein is a minute cell surface glycoprotein whose function has yet to be fully defined. Pathological prions are small infectious glycoprotein particles that cause certain progressive neurodegenerative diseases, lack detectable nucleic acid, and do not elicit an immune response. Pathological prion proteins induce other prions to function in this manner. The gene coding for the normal cellular prion protein is PRP superscript C. The superscript C designates the normal cellular prion protein. PRP refers to the platelet-rich plasma protein of the liquid portion of blood, which contains biologic elements that enhance healing. The abnormal prion protein gene is PRP superscript res or PRP superscript dis. Designations resistant and disease are pathogenic isoforms. Other abbreviated superscripts represent the origin of a pathogenic isoform of a specific disease. This protein is specific to certain neurodegenerative diseases resulting from post-translational modification of the particular cellular prion protein. All diseases believed to be caused by prions to date principally affect brain and neural structures and are untreatable, as well as fatal. A prion is a self-propagating misfolded protein structure capable of contacting its normal protein form, prompting it to also change into an aberrant configuration, often in a chain reaction forming toxic disease-causing prion aggregates. These nerve cell tissue-killing accretions cause neurodegenerative diseases such as Creutzfeldt-Jakob disease and bovine spongiform encephalopathy. Some non-propagating proteins, these are non-self-propagating proteins, possess prion-like protein structure folding properties, having prion-like disease-causing capabilities, as is the case in Alzheimer's disease. Prion-like regions or domains on the protein surface provide for conformational change from one shape to another, as well as these functional disease-causing tissue changes. The presence or absence of particular amino acids create hydrophobic and electrochemically charged regions or domains in a protein structure, causing molecular interactions, enabling this prion-like structural folding. Protein structure in relation to spike protein viruses will be discussed further in a subsequent presentation in this series. Levels of structural organization of proteins illustration. Two, nucleic acid satellite molecules. Nucleic acid satellite molecules, under the influence of a helper virus, function in reproduction and infection. Satellite DNA is a non-coding type of DNA defining repetitive DNA sequences in tandem arrays. A helper virus plays a role in the development of a defective virus by supplying or restoring viral gene activity or enabling protein coat formation, by complementing missing genetic function of a defective satellite virus, resulting in the formation of a functional virus. 3. Viroids. Infectious RNA viroid molecules not associated with or coding for any protein are replicated by host cell enzymes and are causative of several types of plant infection. Viruses exhibit genetic variation and are capable of evolving, as is the case with the various forms of cellular life. Viruses are obligate intracellular parasites, requiring the host cell's biochemical mechanisms to maintain the necessary protein synthesis and sugar metabolism to fuel their survival, function, and replication. The specific amino acid units comprising a protein possess a unique structural sequence determining its configuration and function. Ubiquitins are amino acids that attach to proteins functioning in protein activation, localization, interaction, modulation, inactivation, and degradation affecting cellular processes. Peptides are the constituent elements of amino acids. Enzymes are protein molecules catalyzing chemical reactions without themselves being altered or destroyed in the process. Proteases are enzymes catalyzing the breakdown of peptides and proteins. Cell structure illustration. Ribosomes contain ribosomal RNA, various proteins, and are the site of protein synthesis. The endoplasmic reticulum functions in protein processing and lipid synthesis. The Golgi complex functions in the synthesis of some carbohydrates and the processing of substances synthesized in the endoplasmic reticulum. Difference between DNA and RNA illustration. A nitrogenous base is a nitrogen-containing organic compound possessing base properties, especially those of a purine or pyrimidine. Adenine A, thymine T, guanine G, and cytosine C are the nitrogenous bases of DNA. Adenine, cytosine, guanine, and uracil U are the nitrogenous bases of RNA. Single-stranded RNA containing the sugar ribose functions in the flow of specific genetic information in all cellular organisms and RNA viruses. Cytoplasm is the aqueous solution surrounding the nucleus containing various cellular elements in which most of the cell's chemical activities occur. Uracil is a pyrimidine constituent base of RNA. Uracil binds to adenine. In DNA, its position in base pairing is taken by thymine. Adenine binds to thymine in DNA. Guanine binds to cytosine. The base pairing pattern of RNA nucleotides is adenine uracil and guanine cytosine. Thymine is not a constituent of RNA. As shown in the illustration, by recognizing one DNA strand's base code, the code of the other strand could be ascertained given the known base pairings of adenine thymine and guanine cytosine. RNA is principally found in the cytoplasm of cells, but it is usually synthesized in the nucleus. In contrast to DNA, RNA does not lose one oxygen in its structure. The bases encode for genetic translation for protein and protein synthesis in the nucleus. Nucleic acids and proteins are structurally linked or held together by bonds in chemical compounds. Double-stranded interwoven helical DNA constitutes the primary genetic material of all cellular organisms, as well as DNA viruses, and is found predominantly in the cell nucleus. A nucleoside is a compound found in DNA or RNA linking a purine or pyrimidine base to a sugar. A nucleotide is a compound consisting of a nucleoside linked to a phosphate group comprising the basic structural unit of nucleic acids. Such phosphorus-containing substances, like those involved in cellular energy storage and the structure of bone, are essential to living organisms. A nucleotide is specifically arranged along the double-stranded helix comprising the genetic code for a functional protein in the nucleus. The constituents of nucleic acids adenine, thymine, guanine, and cytosine are the nitrogenous bases of DNA, also referred to as nucleotides. Adenine, thymine, guanine, cytosine, and uracil nucleobases are by definition nucleic acid bases. There is one less oxygen in its structure, accounting for the deoxy prefix of DNA nomenclature. Deoxyribose is a sugar derived from ribose found in DNA. Nitrogen is a constituent of proteins and nucleic acids that is soluble in the blood and body fluids present in all living cells. Purines are carbon and nitrogen containing chemical compounds constituting the building blocks of the DNA and RNA of cells. Pyrimidines are carbon and nitrogen containing organic compounds and are also the building blocks of DNA and RNA. Each DNA strand can be referred to as a sugar phosphate chain structural backbone. Attached to each sugar phosphate unit is a nitrogenous base consisting of carbon and nitrogen rings. The amount of rings comprising the base is determinative of a dual-ringed purine or single-ringed pyrimidine structure. Purines on one DNA strand form hydrogen bonds with the corresponding pyrimidine on the opposite DNA strand, and vice versa, keeping them together. Such base pairing is capable of separation, allowing for replication and transcription, since any existing hydrogen bonds are less strong than covalent bonds. Purines always bond with pyrimidines in the process of complementary base pairing, whereby one DNA strand will exist as an exact complement of the other, yielding this constant ratio of 2 within a molecule of DNA. Viruses express either single or double-stranded DNA or RNA genomes encoding genes. Viral nucleic acids are capable of infecting a host cell, resulting in further virus production. The viruses disassemble, enabling the messenger RNA to carry instructions from the DNA to the cells of the body for the initiation of viral reproduction. A polymer is formed by a union of two molecules. A polymerase is a protein enzyme that catalyzes polymerization. RNA polymerase copies a DNA sequence into a RNA sequence during transcription. In this process, single-stranded RNA having a complementary base sequence to one strand of double-stranded DNA is synthesized in the presence of DNA-dependent RNA polymerases to produce another transcribed RNA strand bearing its genetic information, whose destination is the cytoplasm. Messenger RNA, mRNA, sends the genetic information from the nucleus into the cytoplasm, where translation occurs involving protein amino acids. In the process of translation, a series of codons or triplet bases in mRNA undergo a conversion to an ordered sequence of amino acids of a specific polypeptide chain involving ribosomes, transfer RNA, and various soluble proteins in the process of protein synthesis. Cytoplasmic ribosomes in the presence of transfer RNA bearing a specific amino acid possess three codons or base pairs for the formation for genetically specific polypeptide chain formation involving amino acids capable of producing infectious viral proteins in the Golgi complex, the endoplasmic reticulum, or the cell cytoplasm, leading to the production and proliferation of novel viral particles capable of leaving the host cell to infect other hosts. Viruses can reproduce, exhibiting genetic continuity, as well as the capability of permanent transmissible change of genetic material, resulting in gene mutation. Besides mutation, viral genomes also undergo genetic change by recombination, whereby the naturally occurring change in the structural order of DNA results in novel viral forms. Such gene changes and reassortment can result in the emergence of novel infectious viral strains, capable of causing localized or widespread morbidity and mortality in animals and humans. The coronavirus refers to any of a group of disease-causing RNA viruses of the Coronaviridae family. The genetic code of COVID-19 is adenine cytosine uracil guanine. Observed structural mutations of COVID-19 generate a high rate of uracil residues, resulting in neighboring uracil-uracil pairs that mutated from an original cytosine uracil and uracil cytosine gene sequence. This comprises the mutational profile of the apolipoprotein B messenger RNA editing enzyme catalytic polypeptide-like APOBEC human protein that degrades viral structure, leading to the mutation of viruses, including COVID-19. It edits mRNA deaminating cytosine to uracil, resulting in a stop codon and shortened protein, providing for protein diversity. Misregulated APOBEC enzymes are a source of gene mutation in numerous kinds of cancer. All organisms mutate in what is usually a random process resulting from transcriptional mistakes in DNA copying. COVID-19 gene mutation may not be a random process. This mutation, as regards infected human beings, may comprise part of a defense mechanism to degrade the virus in a process yet to be fully defined. Natural selection involves the survival of the fittest, whereby an organism can adapt to its environment for the purposes of reproduction and long-term survival, espoused by the biologist Charles Darwin in his theory of evolution. This is the means by which the virus is believed to be capable of combating this process of mutation. It has been speculated that if COVID-19 would eventually mutate into a novel durable form of the virus, any acquired immunity to the previous viral form, as well as existing vaccines, would not be specifically protective against infection. Sudden acute respiratory syndrome associated coronavirus, SARS-CoV, is the etiological causative agent of severe acute respiratory syndrome, SARS. This infectious respiratory coronavirus syndrome is characterized by breathing distress, dry cough, fever, headache, and body aches, as well as possible life-threatening complications that will be discussed further in this presentation series. Whole cell genomics can also play a role in susceptibility to severe viral infection. In the case of the coronavirus disease 2019, genetic studies of patient blood types determined by the ABO gene suggest that O blood type individuals may be more resistant and less likely to test positive for the virus than those having blood types A, B, or AB. The four main blood types are determined by the red blood cell group A and or B antigenic sugars that are present. Type O possesses no A or B antigens. Rh positive or Rh negative rhesus protein antibodies are respectively present or absent in a particular blood type. Red blood cells transport oxygen to and carbon dioxide from body tissues. Certain viral antigenic proteins may be better recognized by the immune system of type O COVID-19 patients, providing for a comparatively better immunological response and resistance to severe infection. Type A patients may be comparatively more at risk of developing severe infection, with relatively little differences in susceptibility observed amongst patients having blood types other than O. The reasons behind these observations as they relate to morbidity and mortality have yet to be fully elucidated. Proceed to part 2 of 5.